The overall goal of this procedure is to identify metal oxide nanoparticles in histological samples by darkfield microscopy and hyperspectral mapping. This is accomplished by first identifying a region of interest in a positive control sample containing nanoparticles and capturing a darkfield image. The second step is to create a hyperspectral image or data cube of the region of interest using the hyperspectral camera. Next, a reference spectral library is created from the positive control data cube, which is filtered against a negative control to remove any false positives. The final step is to use the reference spectral library to map against experimental samples to identify and locate nanoparticles of the same composition. Ultimately, enhanced darkfield microscopy and hyperspectral mapping are used to identify metal oxide nanoparticles in histological samples in a more rapid and less resource-intensive manner than other techniques such as electron microscopy.
This method offers several advantages over conventional nanomaterial imaging and characterization techniques such as electron microscopy, in that sample preparation is typically minimal and non-destructive, image acquisition and analysis is less time and cost-intensive, and it can be applied for nanoparticle imaging in a variety of matrices.
Individuals new to the method will sometimes struggle because creating the appropriate positive control samples and reference spectral libraries is both challenging and time-consuming. But once those initial steps are completed, experimental samples can be assessed pretty quickly.
While this method can provide insight into cutaneous penetration of nanoparticles, it can also be applied to other biological samples, such as bronchoalveolar lavage and blood smears, as well as non-biological samples, such as filters used in occupational exposure assessments.
Demonstrating this technique will be Pilar Sosa, a medical student from my laboratory.